Now, I would like to request Mrs. Pallavi Bhosle ma'am to please felicitate Dr. Nandanwar sir with a book and a shawl. Thank you ma'am for that warm welcome. Now, it gives me a great pleasure to introduce Mrs. Pallavi Bhosle ma'am, who is an associate professor and head of the pharmacology department at Srinath College of Pharmacy, Chhatrapati Sambhajinagar. With over 10 years of academic experience, she has published more than 13 research papers in various national and international journals, primarily in the fields of neurology and neuroscience. She also holds an MBA degree in human resource management, along with one Indian patent published, and was honored with a Global Faculty Award in 2022. So with this brief introduction, I would like to invite Pallavi ma'am to please come and introduce our resource person, Dr. Nandanwar sir. Please ma'am.
Good afternoon, everyone. It's my privilege to introduce Dr. M.D. Nandanwar sir, who has been a significant personality in the field of pharmaceutical science for more than 2 decades. Dr. M.D. Nandanwar is a distinguished scientist, Vice President of Preclinical Safety Assessment at Wokhart Research Center, Chhatrapati Sambhajinagar, with over 25 years of experience in drug discovery and toxicology. He has made significant contributions to the development of novel anti-infective drugs. His expertise spans preclinical toxicology, safety pharmacology, regulatory affairs, and investigational drug development, making him a key figure in pharmaceutical research. Dr. Nandanwar has played an instrumental role in successful filing of 6 investigational new drug applications, including 4 US IND approvals, with 2 more filings planned in near future. His leadership in toxicology and safety pharmacology has been critical in evaluating and optimizing the safety profiles of new chemical entities, leading to the development of promising drug candidates. He has also been actively involved in designing preclinical safety studies, including acute, subchronic toxicity, phototoxicity, mutagenicity, teratogenicity, and neurotoxicity assessments. Currently, Vice President at Wokhart, he is responsible for establishing and managing the toxicology infrastructure, leading cross-functional teams in microbiology, pharmacology, pharmacokinetic, and toxicology. His efforts have significantly contributed to the advancement of several NCEs, with MROC and MROC-O being successfully launched for the treatment of soft tissue infections in India. Apart from his corporate contributions, Dr. Nandanwar is recognized GLP, that is Good Laboratory Practice Inspector, for the National GLP Monitoring Authority, Government of India. His expertise in regulatory toxicology and compliance has led to his close involvement in interactions with regulatory authorities such as US FDA, European agencies, Chinese regulatory bodies for the approval and clinical development of novel drug candidates. He has extensive international collaborations. He has worked with top contract research organizations, that is CROs, for conducting GLP toxicity and pharmacokinetic studies. He has also undergone specialized training in cancer research, ultrastructural pathology, and biochemical pathology at German Cancer Research Center, Germany. Dr. Nandanwar is an active researcher and thought leader, having published numerous research papers in reputed journals and participated in international conferences, including those Society of Toxicology, American Society of Microbiology, and the Indian Pharmacological Society, that is IPS. His research contributions include evaluating drug safety, exploring toxicity mechanisms, and advancing new methodologies in toxicology assessments. He is life member of several professional organizations, including Society of Toxicology India, Indian Society of Veterinary Pathologists, Indian Journal of Environmental and Toxicology, Worldwide Fund for Nature. With this impressive career spanning over 2 decades, Dr. M.B. Nandanwar Sir continues to be driving force in preclinical drug discovery, safety assessment, shaping the future of pharmaceutical research with his expertise, leadership and dedication. We are truly fortunate to have you, sir. And now I request sir to please show the details of his expertise with us. Thank you, sir. Thank you.
Hello. Thank you, Pallari ma'am, for nice introduction. First time I came to know that this much work I am doing and I have done this work. Over the 2 decades, I never realized that this much work I have done. Thank you for reminding me. Once again, thank you for the management for inviting me for this lecture. There is already presentation. Stay tuned and awake. So this is very important. This is not mostly I will cover critical assessment of, but the major focus will be on the toxicology assessment of any new chemical entities. How the compound gets screened, the safety is assessed of the compound for the human consumption. So what are the different stages of studies, what are the different type of studies I will cover during this presentation. And short briefly I will cover phase 1, phase 2 and phase 3 clinical trial, how this compound getting screened in the human trials in phase 1 and phase 2 and phase 3 studies. And then what are the regulatory requirements, how FDA is to be handled, what are the documents are required for filing the NDA and marketing. So I will cover from A to Z. So be attentive. So next. Okay. Okay. So I will start with the toxicology. What is toxicology? See, toxicology is a very different field. It is not being taught in any university as a subject of toxicology, at least in India. So toxicology is an amalgamation of many subjects, like pharmacology, biochemistry, pathology, physiology, chemistry. Many subjects come together, or many experts come together, and they form the toxicology assessment. So because one person cannot assess all fields, or cannot be expert in all fields, like in the pathology. So pathology somehow covers many subjects. Like field, without toxicological assessment, drug human consumption. So it is a great opportunity for, particularly for B.Pharm and M.Pharm students, because just now we discussed in the office, this field, M.Pharm or B.Pharm field is such a field that it covers the many subjects like chemistry, pharmacology, physiology, pathology. So many subjects they cover. So M.Pharm students or B.Pharm students can go in any field, even in the regulatory affairs. So what are the reasons for drug failures? Why they in the during research failure phase, why the drugs get failure? So there are major three reasons are there. Sorry. So 80% drug can be detected in clinical phase. So if you are not enough alert or enough expert to understand the aspect of compound in clinical stage, the compound is bound to fail in the clinical stages. So you need to have very good expertise, good experience to assess the compound in the clinical stage itself. Clinical stage means animal studies. Before clinical means preclinical. There is all animal studies. So if you see this, the compound largely fails in pharmacokinetics, sometimes lack of efficacy, and animal toxicology. So pharmacokinetics, there is no need to explain what is pharmacokinetics. Pharmacokinetics means when you give drug to the animals or human, how much it appears in the circulation or how much appears in the tissue level, that is called pharmacokinetics, because drug has to reach to the target. Sometimes whatever, like in case of antibacterials, if your infection is in the brain, like condition like meningitis, so drug has to reach to the brain. Drug has to cross the brain blood-brain barrier to reach the brain and kill the bacteria. So that is called pharmacokinetics, the tissue level pharmacokinetics we call it. And there is a vascular pharmacokinetics, that is a vascular component in blood. So in that case also, your drug may be very efficacious in vitro model, like in plates or some in cell lines. Because cell lines assessment is a very isolated experiment. But when you give to the body, that drug has reached to the circulation, target. So that is called pharmacokinetics. So many drugs get failed in pharmacokinetics, then lack of efficacy. Again, same thing. Your compound may be very active in in vitro models, like it will be binding to some enzymes or targets. But when it goes to the body, really it is binding or not. So that is called lack of efficacy. If it is not binding, then your compound is useless. Then animal toxicology. Again, same thing. If the compound is toxic, there are different stages of studies in toxicology. Like from single dose, repeated dose, like 20-day study, 14-day repeated dose study, then 90-day study, then reproductive toxicology is there, then xenotoxicology is there. So many kind of studies you have to do and then understand whether your compound is safe or not. If your compound is safe at enough safety window, then only that compound can go in the human beings. So because once you complete all the toxicological package, that complete package with the conclusion, we have to submit to the regulatory authorities. The regulatory authority has all experts all over the world, they bring out from all over the world and they read and conclude your data whether it is safe or not. So what is the attrition? Attrition is the failure of drug. Why the drugs fail? I told three reasons. Then again, sometimes there is a very complex targets are there. To send the compound to the particular target. Like I said, blood-brain barrier. Blood-brain barrier is a very tough barrier. Hardly few drugs they get brain barrier crossed. Then placental barriers is there. In pregnant woman, the placenta barrier is a big wall. In that pregnant woman, that drug is very difficult to penetrate. So these are all very difficult targets. So sometimes the complex target also kills your drug. Then cost, if the cost is very high, that also kills your project. So you have to work on such a compound that it should be very affordable to the general public or common public. So very costly drugs are not successful drugs, except like orphan drugs. Orphan means very rare condition drugs. So they are very expensive, but patients are very less. So in that case, your cost is, you can have any cost. But drugs like anti-diabetic, hypertensive, there are so many drugs are there in the market. And if you today introduce the drug in the market with a very high cost, there is no use. So there are the different parameters are there for any attrition or failure of drugs. I will not go around it. Then in discovery, see, discovery is normally the chemistry department synthesizes thousands of compounds, millions of compounds. Out of million compounds of one or two compounds becomes a drug. Other compounds get failed in different what we discuss in different areas, lack of efficiency or animal toxicity or target binding issues. So out of millions of compounds, only one or two becomes product. So for that, this journey of discovery, it takes 15 to 20 years. But now it takes less than that because of lot of technology change, lot of lot of literature is available through Google and lot of sci finders and everything. Earlier days it should take lot of 15 to 20 years, but now in 10 or 12 years that can come out. So what is the drug development process? There are 4 major processes are there. One is discovery phase. Discovery phase mostly limited to the chemistry, medicine chemistry, where they identify the post structure based on the standard molecule available in the market. So based on that, they try to modify the structures with side chains and all these things, and they try to evaluate. So there is a lead identification, then lead optimization, and then candidate selection. So that is a part of medicinal chemistry. But also biology also plays a lot of role in the lead optimizations, because whatever they give one structure with certain chemical moiety, and we evaluate in the biology, and we say now this compound is good, there is some activities there, but we need compound with more activity. So then chemists again go back to the lab and they try to apply their brain and do some modification, again they send back to the biology. So this is called lead optimization process. So then non-clinical phase, again this animal studies where pharmacodynamics, pharmacokinetics and toxicology is important. So when that phase is successful, then compound comes to the clinical phase. There again, there are 3 clinical phases are there, phase 1, phase 2 and phase 3. We will discuss later on the next slides. Then last stage is the marketing phase. So there is NDA, new drug application with the regulatory authorities. Then post-marketing surveillance is important, because once you launch the drug in the market, you have to do the post-marketing surveillance. How your drug is getting performed. That data needs to be submitted to the regulatory authority. That is we call this phase 4. Then additional remaining studies, like I have done complete package of toxicology and pharmacology studies and submitted to the authority. And they got approved and got marketed. But what happened suddenly, when it goes in the clinical use, sometimes unforeseeable or some unexpected findings comes in the human use, like what I call is circling movement. Chakkarani म्हणतो ना आपण, circling movement. This kind of one symptoms. These symptoms we have never seen in the animal studies. Even in clinical studies you have not seen. But when it got to the mass uses of human beings, some patients they noticed that there is some circling movements are happening. So now this is a very completely new finding. In that case, again we come back. That report from pharmacovigilance comes to us. That these are the findings are there. Find out what is the reason for this. So that is called additional or remaining actual study related to it. So what are the preclinical study strategies? So compound selection, how we select the compounds for the development. So first is efficacy should be very high compared to the gold standards. Whatever the gold standards are available in the market, because whatever we want to discover, it should be superior than the market compound. So that our efficacy should be very potent. So there are different models depend on the different therapeutic areas. Like in our area, we work on the antibiotics. So our area we see how at minimum concentration the compound should kill the bacteria, toughest bacteria, we call the resistant bacteria, compared to the marketed compound. Like what we launch the compound Emerald, that is belongs to the chlorophenol class, that is ciprofloxacin, methylfloxacin. So our standard was oxyfloxacin that time. So we always wanted to have our compound better than the or good than the oxyfloxacin. So in that case we see the MIC, that is minimum inhibition concentration. So in that case, if the oxyfloxacin inhibition concentration is 0.5, so ours should be 0.1 at least. So that superiority should be there to further progress the compound. That is called efficacy. Then selectivity. This selectivity area is somehow, I never worked on this area, but this is the receptor binding or ligand binding जो होता है इसमें. In that area the selectivity is important. Like anti-hypersensitive drugs or anti-diabetic drugs. In that case the selectivity is very important. Then again I explained pharmacokinetics is very important aspect for any candidate selection. And of course toxicology is very important. It's a pivotal or vital area of any bacterial or any therapeutic area. Then how to build the confidence in safety assessment? So confidence in safety assessment, manage attrition, means don't try to synthesize compound aimlessly. Synthesis compound in the fewer number, but synthesis mindfully. After studying structural activity relationship and then synthesize the compound. Then you should have driving success, team spirit, objectives, and then preclinical studies. These are the major factors for the drug failures or drug success. So how to bring the confidence? So you have to do a thorough literature survey. You have to understand what is known and what is unknown. Because in drug discovery, patent is very important. It should be, whatever you have discovered, it should be patentable. If it is not patentable, that discovery is not viable. So select appropriate testing species. This is very important again, because whether for studies I have to select mice or rat or dog or monkey. How I should know? So we have to understand the literature, based on the literature survey. Initially, the same class of compound, how they have studied, what type of animal species they are used, why they are used, what is the enzyme homology, how your compound going to behave. On this, all this basis of literature survey, you can select the testing species. Because if the compound is like in many cases, monkey and humans is very same in terms of physiology and even in the disease conditions also. So in that case, if the compound is toxic or very efficacious in monkey, efficacious in monkey means it's going to work very efficacious in the human. If the compound is toxic in monkey, then compound is going to be toxic in the human. So it is not true in all cases, but in 90% this is true. Then initially going for large number of studies or exposure studies, do some small studies like ALS test. ALS test is a very 24 or 48-hour study where your compound will know whether it is xenotoxic or not. So if your compound is xenotoxic, drop that compound. Don't worry. Don't work on that structure or that code. Same thing in neurotoxicities. Cell lines, some certain cell lines are there, they will give indications whether this compound is toxic or not. Then mechanistic compounds, just now we were talking, now we are going to launch one compound as a Mignap. Brand name is Mignap, it's a purely Indian discovered drug from our company. The first time in India we have discovered and we are launching. So this compound belongs to the ketolide class, microlide, ketolide. So like erythromycin, azithromycin, like oxythromycin, in that class. So these compounds are known to cause liver toxicity. So our first target was this compound should be safe in the liver. So we worked on that, that our first focus before going to the any studies, first compound is to come to me. Whether this compound is liver toxic or not, first then only we proceed it. So that strategy we applied, and we found somehow this compound is very safe. And that is the only compound in this class will be launched all over the world. In this class there is no compound on ketolide. There was earlier one erythromycin launched by Aventis, but it is failed in the human. Same like apotoxicity, then renal toxicity, there are compounds like gatifloxacin is a known kidney toxicity. Again, same thing, how to manage attritions or failure, we have to identify risk at the very early stages. So we can kill the compound instead of investing more money in that or time. Then early termination compounds due to unacceptable adverse events save resources. Any compound, not only adverse events, even in the target identifications or early efficacy, which compound is not good, clean. And then prepare a document. Prepare a summary why this compound failed. So this will be a learning document for the junior people or some other people are joining later on. So this will guide you why this compound got failed, and we have a record. Again, this we have to, in toxicology and pharmacology, we have to identify the targets and we have to validate. Without validation, compound, nobody is going to accept the mechanism of action. That is a proof of concept we have to explain. Like GLP-1 or in this antidiabetic GLP-1 analogs and all. So we have to, establish the mechanism and show that this compound is GLP antagonist and antagonist or not. So like this in antibiotics also we have to explain how this bacteria is killing this compound. Like it is cell wall breaking or it is inhibit the protein synthesis. So these are kind of lot of mechanisms are there you have to explain and establish and then propose to the regulatory authority if this is the mechanism of action of this compound. So these are all target validations kind of work. Then what are the type of toxicities we do it? So this is a type of toxicity. What kind of toxicity is required for this particular class of compounds? Like in antibiotics, we don't need carcinogenicity study. Carcinogenicity study means whether this compound is causing tumors or not in animals. So if this compound is causing tumor in the animals, it may cause tumors in the human. So these studies are not required in antibiotic, but these studies are required in anti-diabetic or long-term, life management diseases like diabetes, hypertension, or blood pressure. Then what is the target organ? Toxicity study will give you what is the target organ. Like I explained in this pediatric class, that compound is known as liver toxicity. So this information will be given to the clinical investigator that this compound may cause this kind of toxicity. So in clinical studies or in clinical practice, the doctor will be aware. When patient comes with a liver problem or some hepatitis or some cirrhosis or some problem, so that doctor will be aware that no, you should not give this compound to that patient. So this information we derive from the animal studies. Then dose dependency means what? High dose, mid dose, low dose. So high dose give more toxicity, mid dose gives less toxicity, and low dose should not be toxicity. So like this, dose dependency we establish. And again this you have to correlate with the pharmacokinetics or toxicokinetics. That is AMC, Cmax and Walmart distribution and other things. Then reversibility or reversibility. This is also a very important factor. Like if some compound has caused toxicity in case of liver, in liver. So whether this toxicity, whatever this compound has caused, whether it is reversible or not. So how to understand that? So like the protocol is 14 days treatment is there. So we have given the 14-day treatment to the animals. And at the end of 14 treatment, we have sacrificed the animals on day 15. And we have seen some changes in the liver enzymes and histopathology. But we have to maintain one group. That group will receive treatment up to 14 days and another 14 days will be treatment free. So in 14 days that treatment free, whether these changes are reversed or not, that we have to see. So if the toxicity is reversible, means you have some hope that compound can be progressed for the further development. So there are the kind of studies required. Safety pharmacology, there is CNS safety, then GI safety, cardiac safety. We need then exposure data like again pharmacokinetics in terms of VOC, Cmax. Then single dose toxicity is now we call it an acute toxicity where you have to establish at the maximum dose where 50% animal dies. So we know at this dose 50% animal dies means this compound cannot go beyond that. So whatever the studies are there we have to do, below that dose we have to do. And acute study also we give what kind of toxicity occurs in the animals at higher exposure. Then local tolerance like in the intravenous formulations or intravenous development. When you give the intravenous to the human, sometimes what happens, the phlebotomy is when we insert the needle or cannula, it goes sometimes upward. Patients shake the hands or something like because of irritation. So that needle get detached from the vein and get go in the subcutaneous area. So sometimes it causes the inflammations, redness or swelling. So that also you have to establish whether when the drug goes to the subcutaneous area, whether it is toxic or not. So that is called local tolerance. Then again this genetic mutation, there is mutation and co-aggregation, which is kind of genetic toxicology. Then repeated dose toxic studies, here normally there are many protocols are there. 7 days repeated, 14 days repeated, 20 days repeated, 90 days repeated dose. So you have to dose animals continuously for these much durations. And then after the end of duration, you have to sacrifice the animals, urine examinations, complete blood examinations, that is CBC, and then in biodiversity parameter, almost 42 parameters you have to establish: liver percentage, kidney percentage, lipid profile, electrolytes, all these parameters are there. Then organ weights, then histopathology, gross pathology, histopathology. So entire data you have to pull it together and then draw the conclusion. So for example, I am telling you, at the end of treatment of 20-day study, I have seen some elevation in the ALT. ALT is the liver specific enzyme. When the liver parameter of this ALT increases, so So normally say there is some liver issues there. Then what I had to then correlate this liver enzyme relation with the organ weight, liver. What is the gross pathology of liver, whether the weight is increased or not, then what is the histopathology of liver. So if there is no histopathology, there is no organ weight change, there is nothing changed, only ALT is increased. So I don't call this compound is liver toxicity. These are some transient changes. So that changes should correlate with multiple parameters. That's why in medical practice, sometimes in sonography, some things they see, they send for the further confirmation like MRI or some biopsies. So these are the stages of confirmation. So likewise, we have to understand this or draw the conclusion based on the multiple parameters. Then long-term toxicity I explained up to 90 days. Then the reproductive toxicity is something I will explain in brief protocol afterwards. There is a three kind of reproductive toxicology. It's a male and female fertility studies. Then teratology studies. Teratology means when the drug used to pregnant animals or pregnant women. The baby is growing in the womb of mother after delivery, whether it is causing any structural damage to the baby or not. Like tilted legs, some big head, big nose, something is or structural anomalies. So this we call it teratology changes. Then CARSI studies normally we don't do. These are very expensive studies and these 2-year studies. You have to dose the animal at least 18 months, and after 18 months you have to sacrifice animals. Almost we require 2.5 to 3 years to draw the conclusion whether this compound is causing tumors or not. But these studies are extensively required for life management disease like hypertension and cholesterol management drugs or antidiabetic drugs. But in our area it's not required because antibiotic mostly are prescribed only for 14 days, not more than 14 days. But most of the antibiotics not more than 5 days. Then whatever the studies you are planning or executed, what that study should give to you, what measure the study should give. It should give meaningful and interval data. The data should not be confusing. Confusing means like, suppose you have 3 animals in one experiment. 2 animals have given some changes, 1 animal has not given change. So how will you draw the conclusion? So there are both ways of conclusion. One person will say, no, 1 animal has not shown anything, only 2 animals showed. But one person will say 2 animals showed, 1 person has not showed. So these are very conflicting results. So this kind of changes you have to avoid. That for that you have to have a large number of animals to draw the proper conclusion. Then it should be a clear guidance, whatever the study you have done, it should be clear guidance for the further development, whether this compound should be proceed ahead or not. Then that study, all animal studies will form a very basic foundation for the drug development. It will guide you whether this compound will be successful or not. Then these are the initial studies required for the compounds. Again, receptor activities, then initial PK. On that basis, you can again decide whether compound can go ahead or not. These are very few studies and very short-term studies. In this model also, we can decide whether compound can be developed or not. And these are the typical studies I will explain you. What are the studies required for the safety assessment? This is the ADME or PK studies, where absorption, distribution and material excretion were studied. This study mostly for hours or days, where you understand, as I explained in the PK, once you give the compound to the animals, how much compounds, once you give oral or intravenous, how much compound goes into circulation and how much compound goes to the target and how long it stays in the body. That is also important, because how we decide once a day or twice a day or thrice a day treatment, it is based on the residential time of the drug in the body. If the drug stays longer, then your treatment interval will be longer. So if the drug is clearing very fast, you have to give the drug 2 times, 3 times, 4 times. So that is PK will guide you. In septicology, I explained cardiovascular, respiratory and cells. And telemetry. Telemetry again is normally we do in dog and monkey. This is a telemeter set dogs that transduce input in the animals, and we have to do in conscious dogs. There all cardiac parameters will be monitored. Here again we require two species in one guinea pig, rodent or non-rodent particularly. Rodent means rat, mice, and non-rodent means dog or monkey. See, these are the guidelines is required. This is already guideline available on the website of FDA. So you can just go to the FDA website and find out all details about this study. Then acute study already I explained. These are the parameters normally we see: mortality, clinical observations, weight change, and gross and histopathology. This is a single dose study. Once you dose the animals, we observe for 14 days and see any this compound is causing any changes or not. And this will form a basis for selection of long-term study doses for long-term studies. Then xenotoxicity again. This is a battery of xenotoxic studies. There is a ELF test, there is a bacterial mutation test, reverse mutation test. Then there is a chromosomal aberration. Then micronucleus in human lymphocytes. Then again this chromosomal aberrations, Chinese hamster varices, and then mouse lymphoma. So these are the studies required to understand whether your compound is genotoxic or not. If your compound is genotoxic, it means this compound can cause cancer. So that's why these studies are very important. If the compound is genotoxic, this compound cannot be developed except anti-cancer compounds. All anti-cancer compounds are mutagen. Then subacotoxicity again. Subacotoxicity is 14 days or 28 days. Again, I explained you. We have to dose the animals for continuously for 14 days at 3 graded doses: low dose, mid dose, and high dose. So how to select the high dose? High dose you have to select in such a way that that compound should give at least some toxicity. Mid dose should not give or should give some minimal toxicity, and low dose should not give toxicity. So how to select the doses based on the pharmacokinetics data available from earlier studies. Like your therapeutic UC, in efficacy models you establish that this compound therapeutic UC is supposed to pay. So in toxicology studies, you have to achieve the AUC more than 10, like 20, 40 or 80. So because it's a toxicity studies, so the compound should be, then only you can establish the safety window. Like your efficacy is 10 AUC and you establish safety at 40. So your safety window is 4 times higher than the expected therapeutic exposure. So that's why we have to select the doses for long-term studies. Same like 28 days, 14 days, 90 days, same protocol will be there. And these are the important parameters like mortality, whether animals are dying or not. If they are dying, why they are dying. Clinical observations, what kind of clinical observations, whether clinical observations are related to the GI, related to the CNS, or some eye. So these all parameters you have to understand and you have to relate them with the organ system. Then weight changes, whether this compound is causing weight loss or not. Then clinical pathology, again hematology, biochemistry. Then urinalysis, ophthalmoscope you have to examine whether this compound is causing excessive dryness or some corneal opacity or some retinal damage or not. And then gross and again histopathology. So histopathology you have to do for all organs, starting from nose to tail. All thyroid, parathyroid, pituitary, all organs, brain, kidney, nerve, muscles, bones, joints, everything. Again, here also we have a recovery period, as I explained how to assess the recovery, reversibility of any changes. Same parameters will be our chronic toxicity. Chronic means normally more than 20 days. 20 days study, when you dose animals more than 20 days, we call it chronic study. From 1 to 6 month duration, and again these are the same observation will be there. Then this reproductive toxicology, as I explained, this is a segment 1, we call it fertility, male and female fertility. Here you have to dose the male for initially 28 days, where this spermatogenesis happen, the cycle is 28 days, during you have to dose the animal, and then keep with the female, and see whether their fertility affected or not, based on the sperm examinations and then number of offsprings, any abnormality are happening or not. Then pereology, again this organogenesis, as I explained, this you have to do in 2 species, one is rat and rabbit. This you have to dose animals during the organogenesis period, where after conception, there is a period where organ formation happens. During that organ formation, you have to dose the animals. During organ formation, once you dose the animals, whether this compound is causing any dysfunction of organ formation or not, that you have to study. And then perinatal toxicity, 2 generation studies, where one generation of dose, then there offsprings, again you have to make dose, again you have to meet them. So like 2 generations you have to study, and whether this compound is carrying effect from one generation to another generation or not. That also you have to study. This is a very laborious work and very expensive work. Like fertility index, you have to understand whether this compound is causing any fertility issues. Then again, these are the normal weight change and corpse observation. Corpse observation means their eye opening, then their development, sensory organs, reflexes. This all we have to study. Then case studies, as I explained, we normally don't do, but it is very required for the long-term treatment for therapeutic areas where this compound is causing human formation or not. Then again, toxicology I explained already. So this is the case study. This we study in-house. This compound belongs to fluoroquinolone class, that is MROC and MROC which we launched, that is process in ciprofloxacin class. This compound known to cause joint toxicity, in particular in children. So normally this class of drugs are avoided giving into the children in today's. But normally doctor weigh the risk-benefit ratio and they prescribe it. But it is very risky giving to the growing child. Okay. This is a joint. If you see, this is a control animal. It's a dog joints. We have given this compound to the dog of 1 month old, 14 days treatment. So this joint, see this control, there is nothing, is very clean joint is there. But if you see this formation of blisters here, that some elevated area is there, that is a blister. Here is a blister, that is a blister. So this is called chondrotoxicity. We call it chondro is cartilage. So whatever the cartilage is there in the joint, it causes damage. Then this is the histopathology of that same joint. This is a control. You see this upper level, there is a epichondrium. This is control, high power and low power. See the structure of this cartilage, complete this cavity. And this carbon dioxide got aggregated in one side. It's a sort of kind of indication of inflammations. Another case study we did is the liver toxicity. That class we ketolide. This control liver is the central vein. Complete parenchyma is normal. This is the high power. And see these necrotic changes inside this center. So these are liver toxicity. So these changes will have to correlate with the biochemical parameters. Then liver weight. Then organ weight, body weight. So everything you have to correlate and you have to continue. This is another area we work, that is observability. There is one drug in the market that is called linezolid. You must have heard. So that linezolid cannot be given more than 14 days in human nowadays because of these kind of changes. It sucks up the complete bone marrow. So once the bone marrow is gone, there won't be any erythropoiesis or blood cells formation. So this is the bone marrow complete. There is a huge cellular cells are there in the cavity. You see the bone marrow complete dot vacuolated. All vacuoles are there. There are no cells at all. Then there is thymus. See the cellularity of thymus and the less number of lymphocytes. So these are typical indication of myelosuppressions or bone marrow suppressions. Then what are the kind of studies required? So if your treatment, your proposed treatment is up to 2 weeks, you have to do 2 to 4 week study. If your treatment is up to 1 month, then 1 month study is required. Like this, up to 6 months, then 6 month study is required. So depending on the therapeutic duration in the human, the study design you have to plan. So what all toxicology studies leads to? What they give to you? They give maximum tolerable dose. What is the maximum tolerable dose the animal can tolerate? Maximum tolerable dose means animals cannot die, but it shows some clinical signs. That is called maximum tolerable dose. Then NOL, what is no observed adverse event level? This value is extremely important to decide the dose for the human in clinical trials. So this dose is derived from the long-term studies like 14 day, 20 day and 90 day. So normally we keep 3 doses, low dose, mid dose and high dose. So NOL dose is such a dose where minimal changes are there, but they are not biologically significant. Meaning, like reduce some food consumption in animals. This is not very biologically significant. that dose can be a NOAL. Or suppose some body weight variations there minor, that still dose can be NOAL. Or some minor liver enzymes are increased, but they are during reversal period, still it is NOAL. Then there is another dose, NOEL. In such a dose, there is absolutely no changes. Absolutely no changes. That is called no observed effect level. So, and these doses are NOAL are dose dependent or duration dependent. Because in long term, short term studies, your NOAL dose will be very high. But in long term, dose will be low. Because you are dosing for long term, so your dose could be less, lower NOAL. And for short term, that dose could be high. So these are all doses are important to start the clinical goals. From this NOAL, we calculate based on the body surface area and what could be the maximum safe dose in the human. Then which species give more predictable toxicity to the humans. So as per the literature and my experience, monkey is very congested. Whatever the compound shows in the monkey, it's bound to show in the human. So that's why human toxicity is animal toxicity is monkey toxicity is very important to predict whether this compound is going to be toxic or not in the human. Are you okay? Okay. Should we go ahead? Okay. So as I tell you, this monkey is the best species to predict the human toxicity. On other way, the compound which is safe in two species, like rhododendron, that compound is expected to be highly safe in human. So that's why regulatory authority always recommend to understand or undertake toxicity in two species. One is rodent and one is non-rodent. So normally we do in antibiotics, we do in rat and in dog. We don't do for monkey. Monkey is very expensive study. One study goes almost 6 to 7 crores. So and dog study goes around 3 to 4 crores. So that's why we prepare monkey study. It's a very expensive business and risky business, that discovery. So these are the once your compound is very safe in animal studies, in safety, pharmacology, efficacy and toxicology, then management take a decision whether this compound based on this toxicity profile and basic profile, whether this compound should push ahead for human development or not. So then comes in picture clinical development, that is, and it has 3 stages: phase 1 clinical trial, phase 2 and phase 3. So what is phase 1? Phase 1 is nothing but to understand the PK and tolerability of the compound. So what are the studies are required for phase 1? At least up to phase 1 if you want to go. You need septic pharmacology studies, pharmacokinetics, then acute and suburb studies, some initial genome studies, 1 or 2 studies you require. Initial reproductive studies, you may not require studies, but you may require some structural activity relationship, whether similar earlier compound, whether it's causing any reproductive toxicity or not. Then some local tolerance or specific toxicity means like compound related toxicity or compound specific toxicity. Like acute fluorophenols are known to cause phototoxicity. When you consume the compound and go in the sunlight, we develop skin rashes. So that is called phototoxicity. This is a very classical toxicity of any fluorophenols. Then for phase 2, what are you required? Phase 2 there is normally we require 400 to 500 patients. For this we need complete battery of genotoxicity, complete state of genotoxicity. Then complete phase 1, segment 1, segment 2, segment 3 reproductive toxicology. Then extended repeat dose like from 28 days to then you need 90 day studies and some pharmacokinetic studies. For phase 2 and phase 3, you need a total complete package up to CAR-C studies. So then once you complete the clinical phases, phase 1, phase 2, phase 3, once the compound is safe, then you go for the IND filing. For IND filing means getting permission to undertake the phase 1 study. That is investigation new drug application. That is same in the India and for other countries also. So what they do, clinical key questions from your animal studies: can preclinical studies or clinical bulk be efficiently synthesized or not? Whatever the compounds you have evaluated, whether it is clinical efficacy or not, whether it can be synthesized or not. So if it is not efficacious or not, it's very expensive or it cannot be synthesized, then what is the use? So at this stage you have to take the decisions. Every stage has decisions, go no go decisions. Then manufacture problems or formulation development problems are there or not, whether your formulation whatever development is stable or not. So that we have to understand first before going to further development. Then pharmacokinetics again same thing. Then clinical, what is the PK predicted PK dose and T therapeutic index, and what is the best clinical plan. So how we have to evolve the clinical plan in phase 1, phase 2, phase 3 studies. It all depends on the objectives. Then what they do in investigation new drug application? You have to program your development planning. You have to develop the planning complete. Then there is a special guideline there how to prepare the IND package. So investigation new drug applications. Then once you submit the pre-IND package to the regulatory authority, they arrange the meeting. That is we call pre-IND meeting. Then we discuss all the data we have submitted to the pre-IND. What is the structure? What is the CMC? Then what are the toxicities you are seeing? What is the expected doses in the human? What kind of toxicity you are expecting in human? So these studies are all this data is discussed with the regulatory authorities. Then IND get finalized and then get submitted. So once you submit the IND, they give permission to the phase 1. So then from there phase 1 starts. So these are the data we have to submit, all clinical data to the FDA we have to submit. Then chemical manufacturing control data, that is CMC. Then general investigation plan, how we are going to investigate this drug in the human, that we have to. Then propose clinical protocol studies, how we are going to undertake the studies, what are the doses, how many patients, what are the investigation centers, all your, what are the investigators, clinical institute will be there. So complete plan along with the ethical permissions. Then if we have any previous human exposure with these drugs or with such compounds, that I have to explain to the regulatory authorities. If we don't receive any objections within 30 days, particularly for the US FDA, it means FDA has given the permission. But in the India it is not like that. You have to wait for the permission up to 1 year, 2 years or 3 years. So what is the phase 1? Focus of phase 1 I have already explained. You have to identify the safe dose ranges, but from single dose to multiple dose you have to understand, undertake the studies in the human. And there is single dose studies, particularly in phase 1. How long the drugs stay in the body, this is a kind of pharmacokinetics you have to establish in the human. Now how drug is eliminated from the body. In phase 2, this is a proof of concept phase There we understand whether the drug has efficacy or not, because efficacy is not the objective in the phase 1. In phase 1 only pharmacokinetics and safety. Whether it's in phase 2 efficacy is very important. Then whatever your mechanism you have proposed, that mechanism getting proved in phase 2 or not. Then is your compound is safe or not to the patients, because this phase 1 is normally done in normal individuals, it is not diseased individuals. But phase 2 is always in the diseased individuals. And what is the proper dose? What dose of your medicine can cure the patients? That is called therapeutic dose. Then commercial considerations comes after that. So whatever drug you have developed, do you have confidence that drug will work or not? It will feel medically or not? Be it is differentiated from others or not? Because what existing molecules, if it's 10 rupees and we are going to establish introduce market with 100 rupee drug, if the efficacy of the both the compound is same, who is going to buy your compound? If 10 rupees compound is working, why people will buy 100 rupees compound? That is what commercial I expect. Then again same thing. Whether it is working better than the competitor agent, where is market? Can it be manufactured efficiently or not? If the answer is yes for all questions, then you can go for NDA. That is new drug applications. It is the final stage of drug development. After reviewing this NDA application, you will be getting the marketing permission. So we are already at the NDA stage for one compound. Currently we are writing NDA for the US FDA, and mostly it will be complete by March 8 or April first week. And we are expected to submit complete NDA in the June or July to the US FDA. So this is how the drug development happens in brief. If you have any questions, please feel free to ask.
Sharing your expertise with us today. Participants, please feel free to ask the questions. If you have any questions, please ask.
Yeah, please.
Sir, as you all know, nanotechnology is on the key topics research. Are there any regulatory guidelines for the toxicological assessment?
Yes. Yes. Guidelines almost will be same. What is more important is the kind of doses and the kind of group, and the species selection for toxic studies, whether we require oral or not oral. Otherwise, toxicity assessment normally for any compound on the earth is same protocol. What matters is species selection and dose selection, and how we are going to establish the kinetics for narcotics. Where once you use nanoparticles to the animals, whether they get absorbed in circulation or they get lost in some tissues, or whether they generic tissue damage. So that kind of approach you have to take to establish the safety of this nanoparticle. Because nanoparticles, will be trapping drug in the nanoparticles. So sometimes that nanoparticles were remote drug released in the liver, and the same particle will release less drug in the kidney. So that kind of approach you have to take to understand the toxicity and efficacy of the nanoparticles. Otherwise, guidelines will
clinical screening, and where can I get that opportunity?
see, basically, being a student of pharmacology, you should have basic knowledge of animals first. Like complete physiology and biochemistry of animals, one thing. Then what are basic knowledge required, like complete package, how it works in the in entire body, how the organs act collectively to absorb the drugs or eliminate the drugs in the body. This kind of Really, this monkey is the best species to bait the human toxicity. On other way, the compound which is safe in two species, like rhododendron, that compound is expected to be highly safe in human. So that's why regulatory authority always
No. Thank you.
As we come to the conclusion, I extend my heartfelt gratitude to Dr. Nandan sir and for sharing his immense knowledge and expertise in preclinical drug discovery, preclinical safety assessment, different phases of clinical trials, reasons for drug failure, types of toxicities, safety pharmacologies, post-marketing surveillance, ICH guidelines, his new launches, then investigational new drug filing and other commercial considerations. These things have truly enriched our knowledge, our understanding in this field. Dr. Nandan's contributions to the pharmaceutical research and regulatory sciences is not just inspiring, but it is a testament to power of dedication, innovation and scientific rigor. We hope that this session have provided meaningful takeaways for all of us, especially those aspiring to make a mark in pharmacology, drug development and medical research. A big thank you once again to Dr. Nandan sir for his time, wisdom and to our audience for their active participation. Let's continue to explore, innovate and contribute to the world of science and healthcare. Thank you sir. Thank you.
Now, I would like to request Pallavi ma'am to please felicitate Dr. Nandan sir with a memento and certificate as a token of appreciation and gratitude. Thank you, Pallavi ma'am. Thank you, sir, for your time and knowledge. So, as we wrap up our first day of this enriching conference, I want to express my heartfelt gratitude to all our speakers, participants and organizers for your active engagement and contributions. But before we wrap up, I would like to share some important instructions for tomorrow. So please, all the participants, please report to the registration desk to mark your attendance and ensure you are seated in the seminar hall by 9 a.m. sharp tomorrow morning. Poster presenters, kindly set up your posters by 11:45 a.m. in the designated area. As the first round of poster presentation will begin promptly at 12 p.m. After that engaging session, lunch will be served at 1 p.m., providing a wonderful opportunity to network. Then we will continue with the second round of poster presentation starting at 2 p.m. Finally, participation certificates will be distributed at 3:45 p.m. So please note that the certificates will not be issued without completing the feedback form. And that so please make sure to fill out the feedback form and the link will be shared on the WhatsApp group, and also you can scan the QR codes which are available at the registration counter and also outside the seminar hall. And you have to fill the feedback form of today's also and tomorrow's also. Otherwise you will not get the certificates. So fill out the feedback forms and ensure you receive the certificates. So thank you once again for being here today.